Hello everyone, welcome to the next lecture in our physiology video lecture series. This lecture is titled Circulation Part 2. In this particular lecture, we'll follow up on the knowledge of the previous lecture and also learn about the different type of control and regulation systems that work on the circulation system. So let us start. First off, let us look into the local and humoral control of blood flow by the tissues. The first question that we need to ask regarding regulation of blood flow is what are some of the specific needs of tissues for blood flow? The answer to this includes delivery of oxygen to the tissues, delivery of other nutrients such as glucose, amino acids, and fatty acids, removal of excretory products such as or end products such as carbon dioxide from the tissues, hydrogen ions from the tissues, as well as maintenance of proper concentration of ions in these tissues and transport of various hormones and other substances to the different tissues. Next, we look into what is the difference between the blood flow in different tissues and organs. As is obvious, one would guess that the different tissues in our body as well as different organs in our body will have different blood flow attributed to them. For example, there is a huge blood flow of several hundred milliliters per minute per 100 grams of thyroid or adrenal gland tissue, which can be seen in this table right here, which is around 300 and 160 respectively. So with respect to the weight, the blood flow is very large. On the other hand, we can see that in case of the liver, there is also a large amount of blood flow, which is close to 1350 milliliters per minute. So finally, resting muscle has a very little blood flow of around 750 milliliters per minute, even though muscle constitutes around 30 to 40% of the total body mass. Thus, there is a various different levels of blood flow required for various different tissues and organs, and these need to be minutely controlled by the circulatory system. So another question that strikes us is that why not simply allow very large blood flow all the time through every tissue in the body, always enough to supply the tissue's need, whether the activity of the tissue is little or great. But this is very practically not possible, because to do this would require many times more blood flow than the heart can pump. By controlling the local blood flow in an exact way, the tissues almost never suffer from oxygen nutritional deficiency, and yet the workload on the heart is kept at a minimum. Next, we look into the mechanisms of blood flow control. Mechanisms of blood flow control are divided into two major halves. One is acute control and another is the long-term control. In case of the acute control, this is achieved by rapid changes in local vasodilation or vasoconstriction of the arterioles, meta-arterioles, and precapillary sphincters. And this occurs within seconds to minutes to provide very rapid maintenance of appropriate local tissue blood flow. On the other hand, long-term control means slow controlled changes in flow over a period of days, weeks, or even months. In general, these long-term changes provide even better control of flow in proportion to the need of tissues and are essentially fine-tuned to the specific tissue whose blood flow is being controlled. These changes come about as a result of an increase or decrease in physical sizes and numbers of the actual blood vessels supplying the tissue. So first off, we will look into the acute control of local blood flow and effect of tissue metabolism on local blood flow. As we can see from this figure, it shows an approximate quantitative acute effect of blood flow of increasing the rate of metabolism. So this is essentially a plot of rate of metabolism versus the increase in blood flow. Okay. If we carefully observe, we will see that even an 8 times increase in rate of metabolism will see a simultaneous 4 times increase in the blood flow, which means that the rate of metabolism actually is very much related to the blood flow, and blood flow will increase or decrease with respect to the changes in rate of metabolism. Next, we move on to acute local blood flow regulation when oxygen availability changes. Oxygen availability is also a major factor which controls the level of blood flow into a particular tissue, and we will look into how exactly arterial oxygen saturation affects blood flow. One of the most necessary of the metabolic nutrients is oxygen. Whenever the availability of oxygen to tissue decreases, such as in case of high altitude at the top of a mountain, or in pneumonia where lungs are uh, unable to respire correctly in order to take in enough oxygen, or in case of carbon monoxide poisoning, which poisons the ability of hemoglobin to transport oxygen, or in case of cyanide poisoning, which is the poison which poisons the ability of tissues to use oxygen, the blood flow through the tissues increases markedly. What this means is that the local blood flow changes accordingly in order to maintain a constant level of oxygen concentration. The figure here shows that as arterial oxygen saturation decreases about 25%, the blood flow increases close to threefold. That is, the blood flow increases almost enough to make up for the decreased amount of oxygen in blood, and thus maintaining an exact constant supply of oxygen to the tissues. That is obvious from this particular graph. When the arterial oxygen saturation decreases to 25% of its original value, the blood flow increases 3 times as compared to normal in order to maintain the constant oxygen saturation. Now we move on to various theories which show us how exactly the local blood flow is maintained acutely. So in this part of the lecture, we look into how the acute blood flow regulation is possible via various theories that try to explain it. The first one we look into is the Vasodilator theory, which tells us about the possible role of various vasodilator substances such as adenosine. According to this theory, the greater the rate of metabolism or the less availability of oxygen or other nutrients to a tissue, the greater the formation or rate of formation of vasodilator substances in the tissue cells. So when there is a decrease in oxygen and other important nutrients in the tissues, the tissue cells start releasing these substances known as vasodilator substances. Some of the different vasodilator substances have been suggested, which include adenosine, carbon dioxide, adenosine phosphate compounds, histamine, potassium ions, and hydrogen ions. It has been speculated that the substance adenosine is the most important of the local vasodilators for controlling local blood flow. For example, minute quantities of adenosine are released from heart muscle cells when coronary blood flow becomes too little, and this causes enough local vasodilation in the heart to return the coronary blood flow back to normal. However, a single vasodilator substance is not likely to be sufficient to increase the local blood flow, and hence a combination of vasodilator substances are likely required in order to control the local blood flow acutely. Next, we move on to the oxygen lack theory for local blood flow control. The oxygen is, as we have seen, a very important metabolic nutrient and is also related to vascular muscle contraction. Therefore, in the absence of adequate oxygen, it is reasonable to believe that blood vessels simply would relax and therefore naturally dilate. As oxygen is required for the constriction to occur, so when there is an absence or a reduction of the concentration of oxygen, the blood vessels and the smooth muscles lining the blood vessels may simply relax and therefore naturally dilate, allowing more blood to come into the tissue. Also, increased utilization of oxygen in the tissues as a result of increased metabolism, theoretically could decrease the availability of oxygen to the smooth muscle fibers in the local blood tissues, and this too would cause local vasodilation. The figure that is shown right here shows a tissue unit consisting of a meta-arteriole with a single side arm capillary and its surrounding tissue. So this is the meta-arteriole and this is our capillary. At the origin of the capillary, there is a precapillary sphincter, that is the last ring of muscle around the meta-arteriole, after which the capillary starts. And around the meta-arteriole, there are several other smooth muscle fibers. The precapillary sphincter and meta-arterioles open and close cyclically several times a minute, with the duration of open phases being proportional to the metabolic needs of the tissues for oxygen. The cyclical opening and closing is called vasomotion. So, when there is a decrease in oxygen concentration in the tissue where the side arm capillary runs, the precapillary sphincter as well as the smooth muscle fibers lining the meta-arteriole will dilate and allow more blood to come into the side arm capillary. And thus, oxygen concentration will guide the local blood flow control in this particular tissue. Because the smooth muscle requires oxygen to remain contracted, one might assume that the strength of contraction of the sphincters would increase with an increase in oxygen concentration. Consequently, when the oxygen concentration in the tissue rises above a certain level, the precapillary and the meta-arteriole sphincters presumably would close until the tissue cells consume the excess oxygen. But when the excess oxygen is gone, the oxygen concentration falls low enough, and the sphincters would open once more and begin the cycle again. Thus, when there is enough oxygen and thus probably enough nutrients available to the tissue, then the blood flow can momentarily stop through the capillary by the action of the meta-arteriole smooth fibers as well as the precapillary sphincter. And this goes on till there is a low amount, less oxygen than required, than needed by the tissue. When there is very low concentration of oxygen, the smooth muscle fibers as well as the precapillary sphincter also dilate as oxygen is required for contraction of the smooth muscle fibers themselves. And this dilation then allows more blood flow into the tissue itself. And this blood flow will carry extra oxygen with it. Once this oxygen comes into the tissue, the precapillary sphincter and the smooth muscle fiber contract again and the cycle continues. Next, we move on to long-term blood flow regulation and the long-term blood flow regulation, which is the second type of regulation that is applied on the circulatory system. The mechanism of long-term regulation involves change in tissue vascularity. The mechanism of long-term local blood flow regulation is principally to change the amount of vascularity of the tissues. There is actually physical reconstruction of the tissue vasculature to meet the needs of the tissue, which occurs rapidly within days in extremely young animals. It also occurs rapidly in new grown tissue such as scar tissue and cancerous tissue. However, it occurs much more slowly in old, well-established tissues. Therefore, the time required for long-term regulation to take place may be only a few days in a neonate or as long as months in elderly persons. So, long-term blood flow control essentially changes the entire architecture of the tissues by changing the vascularity or the number and amount of blood vessels that are present in the tissue itself. So, this long-term control regulation can allow the tissue to have greater number of blood vessels than it started with, or decrease the amount of blood vessels that were in order to control and fine-tune the blood levels required by the tissues itself. This total mechanism is known as tissue vascularity. So here also we see there is a role of oxygen in regulation of in long-term regulation of blood flow. Oxygen is important not only for acute control of local blood flow, but also for local long-term control. In the acute control, that is the one that we have just seen. One example of this is increased vascularity in tissues of animals that live at high altitude where atmospheric oxygen is low. So when atmospheric oxygen is low, the blood vessels carry less oxygen, and thus there is a need for excess oxygen at the tissue level. And this guides the long-term regulation via the increase in vasculature of the particular tissues. Thus, increased number of blood vessels will carry more oxygen to the tissues themselves and maintain the normal amount of oxygen concentration. This is an important point that we will look into once we go into further lectures, especially the one at high altitude physiology. A second example is that fetal chicks hatched in low oxygen have up to twice as much tissue blood vessel conductivity as is normally true. So fetal chicks which have a similar effect to high altitude born animals, where when they are hatched in low oxygen, the long-term regulation allows them to have much more blood vessel conductivity in order to contradict or maintain the lower or atmospheric oxygen and maintain normal oxygen levels at the tissue. This same effect is also demonstrated in premature human babies put into oxygen tents for therapeutic purposes. The excess oxygen causes almost immediate cessation of new vascular growth in the retina of the premature baby's eyes. Because oxygen guides the amount of vasculature, so inside the oxygen tent, as there is a lot of oxygen present, the vasculature inside the retina of the premature baby's eyes completely stop. But when the infant is taken out of the oxygen tent, there is explosive overgrowth of new vessels and to make up for the sudden decrease in available oxygen. Often there is so much overgrowth that the retinal vessels grow out from the retina into the eye's vitreous humor, and this eventually causes blindness. Next, we move on to importance of vascular endothelial growth factors in formation of new blood vessels. Three of the best characterized angiogenic factors are vascular endothelial growth factor VEGF, fibroblast growth factor, which is FGF, and angiogenin, each of which has been isolated from tissues that have inadequate blood supply. Presumably, it is deficiency of tissue oxygen or other nutrients, or maybe both, that lead to formation of these vascular growth factors. Next, we move on to humoral control of circulation. Humoral obviously means control via hormones, and we will now look into how hormones are used to control circulation and the local blood flow. First, we look into vasoconstrictor agents or hormones which allow the blood vessels to constrict. The first of these include norepinephrine and epinephrine. When the sympathetic nervous system is stimulated in most or all parts of the body during stress or exercise, the sympathetic nerve endings in the individual tissues release norepinephrine. Norepinephrine excites the heart and contracts veins and arterioles. In addition, the sympathetic nerves to the adrenal medulla also causes these glands to secrete both norepinephrine and epinephrine into the blood. These hormones then circulate to all areas of the body and cause almost the same effects on circulation as direct sympathetic stimulation would. So in case we need a refresher, sympathetic stimulation from the nervous system is the one which is sympathetic stimulation from nervous system is the one which is excited when the body enters into a fight or flight response. That is when the body requires immediate activity, and thus norepinephrine and epinephrine allow the body to go into the highest state of activity by being excited by the sympathetic nervous system, and thus they increase the blood flow and the heart rate, which allows the body to enter this higher state of activity in order to undergo the fight or flight response. So sympathetic nervous system stimulation encourages the fight or flight response. That is the summary from this particular slide. Next, we move on to the next vasoconstrictor agent, which is angiotensin 2. The effect of angiotensin 2 is to constrict powerfully the small arterioles. If this occurs in an isolated tissue area, the blood flow to that area can be severely depressed. However, the real importance of angiotensin 2 is that it normally acts on many of the arterioles of the body at the same time to increase the total peripheral resistance. That is thereby increasing the arterial pressure. So if we go back to our previous lecture, then you will remember that peripheral resistance in a blood tissue is very much related to the flow as well as arterial pressure. So if you remember correctly, so total peripheral resistance is directly proportional to arterial pressure, and thus increasing the total peripheral resistance will also increase the arterial pressure of our body, and thus also help us into going into the fight and flight response. The other vasoconstrictor agent that we will talk about is vasopressin. Vasopressin, also called an antidiuretic hormone. Antidiuretic means that a hormone which decreases the amount of water that is excreted by our body in urine. And vasopressin is an antidiuretic hormone which is even more powerful than angiotensin 2 as a vasoconstrictor. Normally, only minute amounts of vasopressin are secreted, so that most physiologists have thought that vasopressin plays little role in vascular control. However, experiments have shown that concentration of circulating blood vasopressin after severe hemorrhage can rise high enough to increase the arterial pressure as much as 60 millimeters of mercury, which is a huge increase in blood pressure. Vasopressin has a major function to greatly decrease water reabsorption in the renal tubules. Renal tubules means the tubules that are present in our kidney, and back into the blood, and thus it is called an antidiuretic hormone. So we will look into the renal system in upcoming lectures, in which we will look into water reabsorption in the renal tubules. So when the water is reabsorbed back via the renal tubules into the blood flow, then the amount or concentration of water in the urine severely decreases. And this is what the action of antidiuretic hormone is. It decreases the water excreted out in our urine. And vasopressin is, as I said, an antidiuretic hormone. Next, we move on to another vasoconstrictor agent, which is endothelin. Endothelin is a powerful vasoconstrictor in damaged blood vessels. Endothelin is another vasoconstrictor substance that ranks along with angiotensin and vasopressin in its vasoconstrictor capability. The usual stimulus for release is damage to the endothelium, such as that caused by crushing of tissues or injecting a traumatizing chemical into the blood vessel. After severe blood vessel damage, Release of local endothelin causes subsequent vasoconstriction, which helps prevent extensive bleeding from arteries as large as 5 millimeters in diameter that might have been torn during the injury. So, in one of our previous lecture that was on blood coagulation, we have seen that vasoconstriction was one of the ways via which hemostasis is maintained. That is, when an injury occurs and blood starts flowing out of smaller vessels, blood vessels, then huge vasoconstriction in the smooth muscle fibers lining these vessels can effectively block out the damaged part of the blood vessel so that blood does not pour out of the blood vessel and decrease the total amount of blood that is present in our body. One of the substances, the vasoconstrictor substance that is involved in blood stopping the flow of blood out of the body, that is maintaining hemostasis, is endothelin, and it does it by the vasoconstrictor mechanism. And as we have seen that in case of extensive bleeding from arteries as large as 5 millimeters in diameter, vasoconstriction via endothelin can constrict the particular artery, a particular blood vessel, and prevent extensive bleeding. Next, we move on to vasodilatory agents. The first vasodilatory agent that we will look into is bradykinin. The kinins are small polypeptides that are split away by the proteolytic enzymes from alpha to globulins, which are proteins present in the plasma or tissue fluids, by a proteolytic enzyme known as kallikrein. Kallikrein is activated by macerations, which is also known as degeneration of blood, by tissue inflammation, or by other similar chemical or physical effects on blood or tissues. As kallikrein becomes activated, it acts immediately on alpha-globulin to release a kinin known as kallidin. Kallidin is then converted by tissue enzymes into bradykinin. Bradykinin causes powerful arterial dilation as well as increased capillary permeability. Injection of around 1 microgram of bradykinin into a brachial artery of a person increases blood flow through the arm as much as 6-fold, and even smaller amounts of injected locally into tissues can markedly cause a local edema, resulting from increase in capillary pore size. Edema, for just a brief knowledge of it, is the accumulation of fluid into the interstitial region of our blood, interstitial tissues, which can happen due to increase in capillary pore size. Next, another vasodilator agent that we will look into is histamine. Histamine is released in essentially every tissue of the body if the tissue becomes damaged or inflamed or is subject to an allergic reaction. Most of the histamine is derived from mast cells in the damaged tissues and from basophils in the blood. Histamine has powerful vasodilatory effect on arterioles and, like bradykinin, has the ability to greatly increase capillary porosity. That is, increase the pore size of the capillaries, allowing leakage of blood, both fluid and plasma proteins into the tissues. In many pathological conditions, the intense arterial dilation and increased capillary porosity produced by histamine causes tremendous quantities of fluid to leak out of the circulation into the tissues, inducing edema. Next, we move on to nervous regulation of circulation and rapid control of arterial pressure. So after humoral control, or after looking into humoral control of blood circulation, we have understood that how hormones are involved in regulation of blood flow. But as is obvious, hormonal control is slower in nature, and in case local acute rapid control of blood flow is required, we move on to nervous regulation of circulation. Nervous regulation of circulation is the adjustment of blood flow tissue by tissue. Adjustment of blood flow tissue by tissue is mainly function of local tissue blood flow control mechanisms. Nervous control of circulation has more global functions, such as redistributing blood flow to different areas of the body. increasing or decreasing the pumping activity by the heart, and providing very rapid control of systemic arterial pressure. Thus, nervous regulation is involved in the overall control of blood flow than local tissue level specific control of blood flow, which is done by the local acute system. The autonomous nervous system, the most important part of the autonomous nervous system for regulating circulation is the sympathetic nervous system. The parasympathetic nervous system also contributes specifically to regulation of heart function. So if we remember the previous lectures, autonomic nervous system is the involuntary nervous system over which we have no control over. And this is divided into two parts. The first is the sympathetic nervous system. The other is the parasympathetic nervous system. As we were just discussing a bit earlier, sympathetic nervous system is the part of the nervous system which stimulates different tissues in our body in order to prepare for a flight or fight response or an excitatory response from the body. Parasympathetic nervous system acts in the opposite direction. Parasympathetic nervous system stimulates various tissues and muscles in our body in order to be prepared for the rest and digest function or rest and digest activity, which is when the body essentially relaxes. Thus, sympathetic and parasympathetic nervous system work in opposite ends or in opposite ways, but both have various control. Both can control the circulation and the blood flow in different parts of our body. First, we look into sympathetic innervation of blood vessels. The figure here shows the distribution of sympathetic nerve fibers to blood vessels, demonstrating that in most tissues, all the vessels except capillaries, precapillary sphincters, and metaarterioles are innervated. This is because thus a huge part of the circulatory system is innervated by the sympathetic nervous system. The innervation of small arteries and arterioles allows sympathetic stimulation to increase resistance to blood flow and thereby decrease rate of blood flow through the tissues. Innervation of larger vessels, particularly of the veins, makes it possible for sympathetic stimulation to decrease the volume of these vessels, and this can push blood into the heart, thereby play a major role in regulation of heart pumping. Next, we look into sympathetic nerve fibers that are innervating the heart directly. Sympathetic fibers also go directly to the heart. Sympathetic stimulation markedly increases the activity of the heart, both increasing the heart rate and enhancing its strength and volume of pump. We have already seen in detail the innervation of heart via the sympathetic and the parasympathetic nervous system, and I urge you to look back into it if you want to know more about the innervation of the heart itself. Next is the parasympathetic control of heart function and especially the heart rate. The parasympathetic nervous system plays only a minor role in regulation of circulation, as it only innervates the heart specifically. Its most important circulatory effect is to control heart rate by way of parasympathetic nerve fibers to the heart in the vagus nerve. As shown in the figure, the dashed red line from the brain medulla directly to the heart principally is the major source of control from the nervous system to from the parasympathetic nervous system to the heart rate. Principally, parasympathetic stimulation causes a marked decrease in heart rate and a slight decrease in heart muscle contractibility. This is, as we know, because the parasympathetic nervous system innervates majorly tissues around the sinus node as well as the sinus node itself, which is the pacemaker of the heart. And as before, I will urge you to look into the heart lecture in order to know more about the innervation of the heart muscle itself. Next, we move on to sympathetic vasoconstrictor system and its control by the central nervous system. The sympathetic nerves carry tremendous numbers of vasoconstrictor nerve fibers and only a few vasodilator fibers. Vasoconstricted fibers are attributed to essentially all segments of circulation, but some tissues are highly innervated, while others are less so. This sympathetic vasoconstrictor effect is especially powerful in kidneys, intestines, spleen, and skin, but much less potent in skeletal muscle and the brain. The vasomotor center in the next we look into the vasomotor center in the brain and its control of the vasoconstrictor system. So located bilaterally, mainly in the reticular substance of the medulla and of the lower third of the pons, is an area called the vasomotor center. This center transmits parasympathetic impulses through the vagus nerve to the heart and transmits sympathetic impulses through the spinal cord and peripheral sympathetic nerves to virtually all arteries, arterioles, and veins of the body. So this particular vasomotor center is divided into three parts. The first is the vasoconstrictor center or vasoconstrictor area, located bilaterally in the anterolateral portions of the upper medulla. The neurons originating in this area distribute their fibers to all levels of the spinal cord, where they excite preganglionic vasoconstricted neurons of the sympathetic nervous system. Next, we move on to the vasodilator area, located bilaterally in the anterolateral portions of the lower half of the medulla. So bilateral means on these sides and lower half of the medulla. This portion shows a blown up picture of the medulla. The fibers of these neurons project upward to the vasoconstricted area itself, and they inhibit the vasoconstricted activity of this area, thus causing vasodilation. Hence, the vasodilated area does not innervate or does not have nerves that innervate the whole body directly, but instead it inhibits the vasoconstricted area in order to produce dilation. Third is the sensory area located bilaterally in the tractus solitarius in the posterolateral portions of the medulla and lower pons. The neurons of this area receive sensory nerve signals from the circulatory system, mainly through the vagus and the glossopharyngeal nerve. And output signals from the sensory area then help to control activities of both the vasoconstrictor and the vasodilator areas of the vasomotor center, thus providing reflex control of many of the circulatory functions. Next, we move on to control of heart activity by the vasomotor center. At the same time that the vasomotor center is controlling the amount of vascular constriction, it also controls heart activity. The lateral portions of the vasomotor center transmit excitatory impulses through the sympathetic nervous system to the heart when there is need to increase the heart rate contractility. Conversely, when there is a need to decrease heart pumping, the medial portion of the vasomotor center sends signals to the adjacent dorsal motor nuclei of the vagus nerves, which then transmit parasympathetic impulses through the vagus nerve to the heart to decrease heart rate and heart contractility. Therefore, the vasomotor center can either decrease or increase the heart activity. Heart rate and strength of heart contraction ordinarily increase when vasoconstriction occurs and ordinarily decrease when vasoconstriction is inhibited, that is vasodilation occurs. Next, we move on to control of vasomotor center by higher nerve centers. Large numbers of small neurons located throughout the reticular substance of the pons, mesencephalon and diencephalon can either excite or inhibit the vasomotor center. This reticular substance, which are the higher nerve centers that control the vasomotor centers, are described in rose color in this diagram right here, which is in this part. In general, the neurons in the more lateral and superior portion of the reticular substance cause excitation, whereas the more medial and inferior portions cause inhibition. The hypothalamus has a special role to play. The posterolateral portions of the hypothalamus cause mainly excitation, whereas the anterior portions can cause either mild excitation or inhibition. So the hypothalamus can either cause excitation or inhibition. Many parts of the cerebral cortex can also excite or inhibit the vasomotor system. For example, the motor cortex, the anterior temporal lobe, the orbital areas of the frontal cortex, the anterior part of the cingulate gyrus, the amygdala, the septum, and the hippocampus. Thus, these are different parts of the cerebral cortex that can excite or inhibit the vasomotor center and thus indirectly control the blood flow and the circulatory system. Next, we move on to various substances that are secreted by the nerves in order to control or regulate the circulatory system. The first we will look into norepinephrine, the sympathetic vasoconstrictor transmitter substance. The substance secreted at the endings of vasoconstrictor nerves is almost entirely norepinephrine. Norepinephrine acts directly on alpha adrenergic receptors of vascular smooth muscles to cause vasoconstriction. So we have already seen that norepinephrine has a vasoconstrictor activity. The release of norepinephrine from nerves is already something that we have discussed, but via the example of another neurotransmitter, acetylcholine. The mechanism is essentially the same, and again I will ask you to look back into the membrane physiology nerve lecture in order to look into it. The adrenal medulla and the reaction. Next, we will move on to the adrenal medulla and the reaction to the sympathetic vasoconstrictor substance. Sympathetic impulses are transmitted to the adrenal medulla at the same time that they are transmitted to the blood vessels. These impulses cause medulla to secrete both epinephrine and norepinephrine into the circulating blood. These two hormones are carried in the bloodstream to all parts of the body. So now these vasoconstrictor impulse is not directly traveling via the nervous system itself, but also has entered the blood and can affect every tissue that is in contact with the blood. And thus these two hormones now act directly on all blood vessels, usually causing them to usually in order to cause vasoconstriction. But in an occasional tissue, epinephrine causes vasodilation because it has a beta adrenergic receptor stimulatory effect, which dilates rather than constricts certain vessels. So this alpha and beta adrenergic receptor essentially means different types of the same receptor, adrenergic receptor. So if an alpha adrenergic receptor is present, it will act via, it will show vasoconstriction in presence of norepinephrine and epinephrine. But if a beta adrenergic receptor is present, it will show vasodilation. Next, we move on to sympathetic vasodilator system and its control of the central nervous system. It's controlled by the central nervous system. The sympathetic nerves to skeletal muscles carry sympathetic vasodilator fibers as well as constricted fibers. The vasodilator effect is believed to be caused by epinephrine exciting specific beta adrenergic receptors in the muscle vasculature. The principal area of the brain controlling this system is the anterior hypothalamus. Next, we move on to role of nervous system in rapid control of arterial pressure. One of the most important functions of nervous control of arterial circulation is its capability to cause rapid increase in arterial pressure. Three major changes occur simultaneously, each of which help to increase arterial pressure when controlled by the nervous system. The first is first includes almost all arterioles of the systemic circulation to be constricted. This greatly increases the total peripheral resistance, thereby increasing the arterial pressure. Next, the veins especially, but the other larger vessels of the circulatory system as well, are strongly constricted. This displaces blood out of the large peripheral blood vessels towards the heart, thus increasing the volume of blood in the heart chambers. The stretch of the heart then causes heart to beat with a far greater force, therefore pump increased quantities of blood. This too increases the arterial pressure. This point essentially involves the stalling mechanism of heart contraction. Finally, the heart itself is directly stimulated by the autonomic nervous system, further enhancing the cardiac pumping. Much of this is caused by an increase in heart rate. In addition, the sympathetic nervous signals have a significant direct effect to increase contractile force of the heart muscle. This too increases the capability of the heart to pump larger volumes of blood. This contributes still more to the acute rise in arterial pressure. Hence, all of these 3 points are the ones that are used by the nervous system in order to increase or change the arterial pressure rapidly. An essential point here to consider is that an important essential point here we have to consider is that the nervous system affects the arterial pressure. The response is very rapid in nature. An especially important characteristic of nervous control of the arterial pressure is its rapidity of response, beginning within seconds and often increasing the pressure to 2 times normal within 5 to 10 seconds. Next, we look into the reflex mechanism for normal arterial pressure maintenance. There are multiple subconscious special nervous control mechanisms that operate all the time to maintain the arterial pressure at or near normal. Almost all of these are negative feedback reflex mechanisms. And as we know, negative feedback loops mean that if the arterial pressure increases too much, then the system itself will start decreasing the pressure, and in case the arterial pressure decreases too much, it will automatically start increasing the arterial pressure. Next, we look into the baroreceptor arterial pressure control system, which is the baroreceptor reflexes. This reflex is initiated by stretch receptors called either baroreceptors or pressoreceptors located at specific points in the walls of several large systemic arteries. A rise in arterial pressure stretches the baroreceptors and causes them to transmit signals into the central nervous system. Feedback signals are then sent back through the autonomic nervous system to the circulation to reduce the arterial pressure downwards towards the normal level. Now we will look into the baroreceptors in detail. First, we will look into the physiologic anatomy of the baroreceptors and their innervation. A few baroreceptors are located in the walls of almost every large artery of the thoracic and the neck regions. But as shown in the figure, baroreceptors are highly abundant. Firstly, in the wall of each internal carotid artery, slightly above the carotid bifurcation, which is an area known as the carotid sinus, and the wall of the aortic arch. Signals from the carotid baroreceptors are transmitted through a very small Hering's nerve to the glossopharyngeal nerves in the high neck, and then to the tractus solitarius in the medullary area of the brainstem. Signals from the aortic baroreceptors in the arch of the aorta are transmitted through the vagus nerve also to the same tractus solitarius of the medulla. Next, we look into the response of the baroreceptors to pressure. The figure here shows the effect of different arterial pressure levels on the rate of impulse of transmission in Hering's carotid sinus nerve. Note that the sinus baroreceptors are not at all stimulated when the pressure is between 0 to 50 and 60 millimeters of mercury. But just as the pressure rises above this 50 and 60 millimeter mercury mark, they respond progressively rapidly and reach a maximum at around 180 millimeters of mercury. The responses of the aortic baroreceptors are similar to those of the carotid baroreceptors, except they operate in general at around 30 millimeters higher mercury pressure. This is obviously because the aortic baroreceptors are present right at the opening of the left ventricle, and there they are directly affected by the full force of the systole. from the left ventricle. Next, we look into the circulatory reflex initiated by the baroreceptors. After the baroreceptor signals have entered the tractus solitarius of the medulla, secondary signals inhibit the vasoconstrictor center of the medulla and excite vagal parasympathetic center. The net effect of both these are the vasodilation of veins and arterioles throughout the peripheral circulatory system and a decreased heart rate and strength of contraction. Therefore, excitation of the baroreceptors by high pressure in the arteries reflexively causes arterial pressure to decrease because of both a decrease in peripheral resistance and decrease in cardiac output. This is how baroreceptors are help in sensing and regulating the increase in arterial pressure. Next, we also have a diagram showing us a typical reflex change in arterial pressure caused by occluding the two common carotid arteries. Occluding here means that we are blocking the two carotid arteries. This reduces the carotid sinus pressure. As a result, the baroreceptors become inactive and lose their inhibitory effect on the vasomotor center. The vasomotor center then becomes much more active than usual, causing the aortic arterial pressure to rise and remain elevated during the 10 minutes the carotids are occluded. So at this point in time, both the carotids were clamped and blocked. This caused the baroreceptors to not be activated. So in the normal functioning, normal conditions also, the baroreceptors signal to the vasomotor center and have an inhibitory effect. But when the both the carotids are clamped, this inhibitory effect is also gone, and thus the blood pressure rises to a much higher level, which is maintained till the carotids are reduced and the baroreceptors come back online in order and send inhibitory signals to the vasomotor center, which brings down the blood pressure back up. Thus, the baroreceptors do not directly have an effect in sensing the decrease in arterial pressure, but the removal of baroreceptor impulse increases the arterial pressure, and thus they indirectly help in sensing low blood pressures as well. Next, we move on to control of arterial pressure by carotid aortic chemoreceptors. Effect of oxygen lag on arterial pressure. Closely associated with the baroreceptor pressure control system is a chemoreceptor reflex that operates in much the same way as the baroreceptor reflex. The chemoreceptors are chemosensitive cells sensitive to oxygen lag, carbon dioxide excess, and hydrogen ion excess. They are located in several small chemoreceptor organs about 2 millimeters in size. These are known as the two carotid bodies, one of which lies in the bifurcation of each common carotid artery, and usually 1 or 2, 3 aortic bodies which are adjacent to the aorta. So chemoreceptors are numerically much lesser than the baroreceptors and sense the chemical composition of the blood and are sensitive to oxygen, carbon dioxide, and hydrogen. The chemoreceptors excite nerve fibers that, along with the baroreceptors fibers, pass through the Hurins nerve and the vagus nerve into the vasomotor center of the brainstem. Whenever the arterial pressure falls below a critical level, the chemoreceptors become stimulated because diminished blood flow causes decreased oxygen as well as excess buildup of carbon dioxide and hydrogen ions that are not removed by the slowly moving blood. The signals transmitted from the chemoreceptors excite the vasomotor center, and this elevates the arterial pressure back towards normal. However, this chemoreceptor reflex is not a powerful arterial pressure controller until the arterial pressure falls below 80 millimeters of mercury. Therefore, it is at lower pressures that this reflex becomes important to help prevent still further fall of pressure. So this is all we had for today. In this particular video, we looked into the regulation and control of the circulatory system via various mechanisms present in our body. The references for these slides include the chapter 17 and 18 from the textbook of medical physiology by Arthur C. Guyton, the 11th edition. I thank you for listening to this video.